Back from the break that ended up being an extended break. So hopefully you were able to refresh yourselves. We got some comments going on in the chat there about cruising. Since I guess the last presentation we were talking about norovirus on cruises a lot. So welcome, welcome, welcome. Now I want to quickly introduce our next speaker since we already were delayed. I want to jump right in there to continue our theme of the day on norovirus. So our next speaker is going to be Elizabeth Richardson, and she is Division Manager for Coconino County Health and Human Services in Arizona. Her division oversees environmental health, animal management, vital records, smoke-free Arizona, and forensic interviewing. She has a degree in environmental science from Texas State University and has been working in Coconino County for 7 years, and I'm probably screwing that up, those words, sorry about that.
It's
She's
Coconino.
also a registered Coconino. Thank you.
You're
Elizabeth.
welcome.
She is also a registered environmental health specialist and registered sanitarian. So Elizabeth, the screen is yours. Please take it away.
Thank you so much. And thank you all for inviting me. And I am just humbled and awed to be here as I'm seeing over, it looks like 1,000 people that are watching right now. This is just a great opportunity. I want to start out by saying thank you to everyone at the Grand Canyon who has been so lovely to share this and wanted to use this as an opportunity to educate. And thank you to the FDA for inviting me to speak. And really thank you to the whole team that showed up to do this, because this is in no way just me. This was a large group of people who came together to make our parks as safe as possible in the name of public health. So thank you. Can everyone see my screen? And if I am not hearing anything, I am going to go ahead and move forward. So this is about a norovirus outbreak that happened in the Grand Canyon in 2022, just this year. Again, about me, my name is Elizabeth Richardson. And the county that I'm from is Coconino County. And this is a county in northern Arizona that holds the Grand Canyon. The fun fact that I'm mentioning here is that when this happened, I had been just promoted to division manager, and I had been in my position for 3 months. So clearly I needed a lot of help. All right. So this is the dream. It is beautiful. And most of the time it is this lovely. Here we've got a tent camping and a riverboat that would be like we're going to be discussing today. This scene attracts 6 million people to the Grand Canyon every year to hike, camp, and boat on the Colorado River. Again, 6 million people in our part at Coconino County. We do foodborne investigations with the Grand Canyon. We inspect river guide warehouses where they prep for these trips. We do education concerning outback food handling and outback in general, safety in the outback. River guides take tourists into the canyon on riverboat trips that last anywhere from 3 days to 16 days, and sometimes even longer, I'm told. In a normal year, we sometimes will get 3 riverboat trips affected by norovirus. Some years we get none at all. And 2022 would, as I've said here, was not a normal year. It was excessive. So the basics, you all probably already have it today, but just to kind of go over it again, norovirus is a very common gastroenteric illness, vomiting and non-bloody diarrhea. We've got an incubation period of 12 to 48 hours. And large outbreaks are associated with norovirus every year. And the interesting thing is we usually associate this with restaurants and cruise ships and not necessarily with the Grand Canyon or places in the outdoors. And I think that's why this is a really interesting topic. Why is it so difficult to stop? Again, there are infected individuals that shed the virus at a very high concentration. in feces and also in vomit. It only takes a few virus particles to become infected. And this third bullet is going to really be huge here. Not everyone who is infected shows symptoms. Common backcountry sanitation practices, hand sanitizer. There's not always sinks there. Hand sanitizer is not great at killing the norovirus. Water filters used for backpacking do not remove the norovirus. This is also probably key. And what we found out, the virus can stay alive in food, water, and surfaces for a very long time, weeks. Norovirus can spread through the air. Okay, again, most of the education, this piece I've gotten from the CDC shows indoor environments. We really don't think about being in the outback and norovirus, so this is unusual. Another point that I wanted to bring out here is that incubation period of 12 to 48 hours was key in this case. All right, timeline. On April 8th, 2022 at 3:42 p.m., I received an email with the subject, 1 positive COVID-19 case and 7 passengers exhibiting vomiting and nausea symptoms. And this was early in the season, and yet not unprecedented. And it started our norovirus, it started our outbreak in this way. It would go on to affect several hundred people and last the next 3 months. We could not have known this on April 8th. The email said the following: there was a confirmed COVID-19 positive case. The individual was exhibiting mild symptoms and tested positive through a rapid test. And there was suspected norovirus. On this trip, there was 28 passengers, 4 crew members, launch date of April 5th, a trip exchange date. And what that is some folks start at a certain area earlier or even hike down to get onto the riverboat. And some people show up later. Sometimes supplies are exchanged, sometimes people, sometimes new people get on, sometimes crew members get off, and that's what the exchange is. The trip end date was on 4/16/2022. It was a motorized vehicle. All right, this is a very colorful timeline, and I probably won't go over this whole thing because there's some things that I want to elaborate on. I've got this listed at the end. Some of the important things here are that this started on April 8th. We got our first email alert. We have several others along the way, and it officially ended on July 7th, 2022. I do think it's important that it shows some dates on here where the CDC was requested, a team focus of investigate and educate. Very fun for me was on May 23rd. I was actually at an FDA workshop while this was happening and still trying to take messages and notes and answer emails. And they were kind enough to let me indulge me in that while I was in their workshop. But this is actually where the FDA asked me to participate or to do this education that we're working on today. So thank you all very much. All right, I'm going to move forward. The geography of the Upper Colorado turned out to be very important here as it runs through the canyon. Most trips begin at Lee's Ferry, and I'm going to show you a picture of this in the next slide. Lee's Ferry is mile 0 and conclude at Whitmore Wash, which is mile 188. Over the course of 1,888 miles, were some of the areas that we took samples from, and that is a lot of area to take norovirus samples. So here's the area that we're talking about. We're here in northern Arizona, and this is the Utah border. You can see this is generally the area that we're talking about. Lee's Ferry is where most crafts launch from, mile 0. And although this can go longer, most trips stop at mile 188. Of interest will be Phantom Ranch. That is at mile 88, and that is where I will show you some graphs later that we noted that there was an uptick in transmission of norovirus. So you can see that this is a very long riverway with lots of stops on the way. But again, 0 at Lee's Ferry, 88 Phantom Ranch, and 188 miles in total that we looked at during this investigation. Okay, so our outbreak after our first email kept getting larger, and we couldn't track a single source. We got another norovirus outbreak notification on May 6th, on May 13th. They kept on May 9th, we've got one, on May 10th. They were getting closer and closer together. More and more people were becoming infected. It looked like it was unusual. And we were trying to track down, was this from food? Was this simply person to person? Could it be sewage? Contact surfaces? And we started asking questions like, why is this happening so rapid fire? What is making this year different? And we started meeting. Within a few weeks, we had multiple trips reporting noro, and approximately 100 people were affected. I've tried to block out the outfitters here, but you can see the dates, and they were coming in very rapidly. Over here, the number of people and illnesses, 29 on one trip alone here. The exchange location and date, that would become important later. We were trying to find where more infected people getting on, and just what was happening. But at this point, we were just trying to gather data and try and find out what was happening and what was making this year unusual. And then something shocking happened around May 16th. Hikers were reporting norovirus. At this point, we had 11 commercial river trips that reported GI illness, which would be much more than our average of 3. One non-commercial trip, one Grand Canyon research trip was affected, and on this date, May 16th, 4 hiking and camping trips were reporting GI illness to us. And to us, that was shocking because it made this leap from just being something that was on boats to random people in the park were now getting norovirus, and we couldn't understand why. We called the CDC on May 16th, 2022, because we felt like we needed help and this was bigger. And we didn't fully understand. We didn't want to risk public health or the season in the Grand Canyon for people. And we were very happy to get any help we could at this point. Also on the 16th, Coconino County and the National Park Service issued a press release. And I'll refer later to the steps in Standard 5 with the FDA, but these are all things that we should be doing, whether we're on the epi side or the environmental health side. We should be alerting the public, and we did in the state, and we definitely talked about things about letting people know that this was happening in the park. In the event that they wanted to change their vacation plans, they would know in advance. But then also letting people know that it was imperative if they were feeling ill within 70 hours to please don't join the group. Hand washing, avoid sharing food and drinks, ensure water is not only filtered but chemically disinfected. During visits to water features throughout the canyon, do not drink from non-potable water sources. So we tried to get this message out. And we certainly National Park staff started posting flyers, and anyone who came into contact coming into the park was given information about how to be as safe as possible. Okay. Our team, and I feel, well, I think that I'm making an attempt to be funny here. I'm not sure if it's funny. At this time, we all freaked out, and seriously, we all really got to work because we realized this was highly unusual. And I wanted to bring in the names of the team members here because, again, I cannot say enough. I was new coming into my position. This was a huge team effort with people who are working around the clock, trying to resolve this for the sake of public health and for the season in the Grand Canyon. The OPH is public health within the Grand Canyon. Ronan King, Chris Klein, Maria Saeed, Stephanie Campbell, Hasna Karim were all instrumental, all of these folks. And we started having, somewhere around this period, maybe before the 16th actually, we were having twice weekly meetings to update and to stay into contact with each other. But again, after this, it changed in a huge way. I also want to thank everybody who worked with us from Coconino County, the Grand Canyon Park. We had not only our environmental health team working on this, but our communicable disease team, and our epidemiology team. CDC folks were huge, and a lot of the data that I'm sharing today came from them. Laura Calderwood was instrumental in assisting with slides, and just really wanted to help make this the best education possible for people. The state of Arizona, Jolie Weiss and Connor Fitzgerald assisted, and at the state lab, Dr. Gerba, Arizona State Lab, was testing our samples. All right, so in our meeting around the 16th of May, and in the event that this ever happens to you, and I just want to say, I just want to take a moment and pause and say, I could never have imagined that I would be involved in something like this. I'm in a small rural county. I could not have imagined that I would be involved in an outbreak like this, and that it would revolve around a vacation or a tourist attraction in the outdoors. So if any of you out there are thinking this could never happen to me, I think that it could, and you might want to take a look around and think, how could this happen here, and would we be prepared? So one of the next steps that we took was breaking things down into tasks and roles. And the National Park Service had certain roles that they took on, and concessionaire's office definitely had certain roles. Coconino County Health and Human Services, we broke that down even further into our epidemiology and communicable disease and our environmental health area. Communication was very important, and our staff here with within Coconino County is Trish Lees. She was amazing. NPS, the National Park Service, also had their communication team on board. The CDC, we broke down their tasks, as well as the Arizona Department of Health Services. So it became so large that we had to make sure that we weren't duplicating our efforts. And if something like that happens in your area, you would do something in the similar way. Okay, so our hypothesis, what was happening? So here were some of the ideas that we had. Introduction of noro from the community when people came on board at the beginning or at exchanges, highly likely. Person-to-person spread on riverlet rafts, highly likely. At some point, we wanted to make sure, is everything being sanitized? Is this being cleaned after every trip? We didn't know for sure, so we got that word out. Is there contaminated food? although they are using major suppliers, is their prepared food. And we definitely went out to our warehouses and started looking around to make sure what was there contamination that could be happening on the food side. Contaminated drinking water from the river, not adequately chlorinated or disinfected. This would turn out to play heavily, and also would turn out to be highly likely as far as the hikers. Many people take filters into the canyon, and just to let you know, also even the river guides, if they brought down enough water for everyone in advance, it would sink the boat. They have to get water on the way, and they either filter it from the Colorado River. Sometimes they get potable water from water spigots in the canyon, but it's very remote. It's very rustic. Water isn't always there. And so sometimes it's literally scooping water out of the Colorado River and filtering it, but we also need to make sure that it's chlorinated. And so if people were vomiting or defecating into the water after they had become ill, it was likely that people who were not chlorinating that water were getting norovirus in that way. Potentially food preparation issues. Food is being washed with non-potable water, lack of glove use. So it could be that people were washing food with that non-chlorinated water from the river, not suspecting that it was an issue. It could be that the river guides were ill and passing it on that way. Fomite transmission. Could be boats, could be water spigots, and environmental contamination. There are beaches down there that are places where there are waterfalls or water features that people are very attracted to, and that noro could have been left in that way. We wondered if it could be wastewater plants, but Ronan went and checked into that, and everything was functioning normally at the time. But we wanted to bring in everything that we thought could be possible. We focused on education and identification. River guides gave us information on ill people with these kind of forms. We got numbers based on these forms. We in no way think that we got reports of everyone who is ill, and I'll show you the final numbers, but I think that obviously it was much higher than that because we simply did not get these forms from everyone who was ill in the canyon this year. Okay, we checked menus and food sources. So we looked at who was serving what and tried to see if there was any reported suspect foods. We also went into all of the river guides' warehouses to see how they were preparing food and if there was any exposure in that way. We checked potable water distribution systems. There are some areas that you can get water, potable water in the canyon, and it looks something like this. Very remote and rustic, and sometime during the outbreak, these were being washed down several times a day with chlorine and being attended to. Okay, wait, this is the fun part. How do we even know it's norovirus? Samples. You may not be aware that fecal samples need to be taken, and who takes them? We do. This is a very brave soul, Maret Gebhardt, who works with us in communicable disease. And usually we switch off when we need to take these samples in between environmental health, epidemiology, and communicable disease. This just happened to be her turn. And you might notice that there are ammo cans in the background. These have been called groofers by the river guides. And because originally people would just sit on these in the canyon and use them as the receptacle for them to defecate or urinate in, pack it in, pack it out. And so this was the best way, I think, to manage this. It doesn't leak. Once they're full, they are sealed and they pack them out of the canyon. You'll certainly note that Moret is in full gear here. So also, it's important to note that the canyon goes down several hundred feet. And so the elevation changes. And in Flagstaff, where we are, we're at approximately 7,000, 7,500 feet in elevation. And so we have to be very careful when we open these groovers because we never know exactly what's going to happen due to that change in elevation. Also, we ask that the backcountry river guides mark these cans or whatever it is that they're pulling out. When people become ill, if we get 10 or 20 of these back from a trip, we really don't want to go through every one. We actually don't even want to go through any of them. But we ask them to mark these cans somehow so that we'll know the cans that we're looking at when people were becoming ill. This one you'll see has a blue piece of tape on it, which indicates to us that is an infected can. Sometimes they'll just put an X on the top with a marker or chalk, that's fine. So we have to carefully open these up, and then we will take a few samples out of each can. I have some video, but I thought that might be overkill being that we're close to lunchtime. Okay, so again, ground zero, pack it in, pack it out. Someone was telling me in a state meeting that this was actually an improvement and that sometime over the history of tours in the Grand Canyon, they actually didn't even have these groovers. And so it took some outbreaks early on in the history of tourism to make people recognize that they needed to pack it out. But this is what it looks like in the field, Lysol can included. They put a toilet seat on it, and then they close these up when they're full. So certainly we conduct education for our river guides, and they understand that if people do become ill, it's important to try to keep things separate. So if they're on a trip where people have become infected, it's highly unlikely that they're going to airlife people out of the canyon, and they're going to have to remain in a group until the end of the trip. And so the best they can do is trying to keep them as separate as possible. They'll try and have separate groovers, keep them separate in the boat, and just have people eat separately and keep things as clean as possible. Masks are always an option. Okay, how many samples did we take? So about 60 plus. And so you'll see over here, some of them did show up to be negative, but many did also show up to be positive. Education and prevention, symptom screening prior to getting on rafts, no exchanges occur if there's illnesses on boats, separation of sick and healthy people to the extent of possible, clear direction on need to chlorinate drinking water and not just filter, enhanced environmental cleaning as possible with 1,000 to 5,000 ppm chlorine, certainly exclusion of boats for one round to allow sanitation, heavy, deep cleaning, hand washing, glove use for food preparation, precautions around food preparation, more frequent cleaning of bathrooms in Lee's Ferry and any place really that was public. Flyers were posted on hand washing and norovirus in public areas. Okay, this isn't, I don't want to get into great detail here, but the overall premise is that there are at least 5 to 6 trips during the summertime that launch every day. And so even as challenging as things were during the summer, looking at the amount of daily activity, I think that we did pretty well as a team to keep this reined in as much as possible. Certainly we gave out this information on how to protect your guests to the river guides and anyone in the tourism field. We found some suspects, mile 88, Phantom Ranch in the water. This is how someone would filter their water, and they would need to chlorinate as well. Going back, and I wanted to show you this before we get into some slides. We will see that we had a lot of people here at mile 71, 81, 88 become ill, and that would give them plenty of time to be around other people with norovirus and become infected. Updating the hypothesis again, people were filtering water but not sanitizing it with chlorine. Around Phantom Ranch, there was a spike with human contact and fomite contact. The epi curve, we're seeing some of the most cases around May 4th, but really after this date, it was heightened until the end of May, really. Timeline, we could see here in this gray area, the gold is when we would get our first infections. You can see that people were on the trips often for days before that first infection started. Location and timing of the first illness, onset by group, lots around these exchange dates. So lots of on and off and exchanging, new people coming in, possibly with noro. And this is the area where we're seeing the river mile and where we're seeing that biggest number of infections around mile 80. And distribution of illness along the river, same, really the same, right around mile 80. Attack rates, it's interesting to look at percentages here. This is crew, passengers, and overall. One of the things that we found out about the crew is that they often do not sanitize their own water because they go on so many trips a year and they do not like the taste of it. And so I think that we all learned a lesson this year that it's just got to be a must. They have to, passengers higher in this trip G, and overall this trip G. Demographics, a bit more males than females, and the 60 to 64 range was affected the most. I have here sanity check. How can this happen? I think that after COVID, we all know how it can happen, but especially here, I think it's worth mentioning that this is the trip of a lifetime for folks. They have spent thousands of dollars coming from other continents to the Grand Canyon or other tourist destination. And for airfare, it's a dream of a lifetime. They've told their friends, they've posted on social media, and they've spent thousands of dollars on this rafting trip. And if they feel a little fever or a sore throat just before, I think that they're going to try and push through. And so this is why I believe that even though the river guides and companies call days before and ask if they're well. And even though they ask right before the trip and they get on the boat, I think this is why it continues to happen. We did get some press on this. The Washington Post picked it up on June 29th, 2022, and the Daily Beast. These are some of the headlines. And I wanted to go over some of the FDA Standard 5 key points for anyone who's dealing this in as a county or anyone really in the future. Have a standard operating procedure for FBI outbreak, a list of agencies to call, a MOU with epi investigators if you don't have them at your entity, program guidance and how to collect information and specimens, when to contact the CDC, lab support and documentation, and when to contact media and law enforcement. There's more to Standard 5, but I feel like these ones were issues that really jumped out at me. Summary, we had 116 cases from the National Parks Service case reports, 222 reported cases total from April through July, 136 from commercial rafting trips, 31 hikers, 55 private rafting trips. Again, I suspect that it was much higher, but these are what we got these case reports from. Where did it come from? Ultimately, it was inconclusive. There was not a single smoking gun, person-to-person transmission, certainly, campsites and water faucets, perhaps, water filtered but not chlorinated, very likely. Here is, again, that timeline and these just bunches here of lots of reports from this April 21st through May 10th, 5 trips in one day on May 10th were affected. May 16th being the day the hikers, we got the report on that. CDC came in and helped with the CDC and National Park Service. The team focus was to investigate and educate, split us up into team tasks, do these epi case control studies. We had a town hall with concessionaires and ended right around July 7th is when we called an end to the outbreak. Thank you so much. And again, I want to thank the outstanding team and how kind they were to me, that this was my first major outbreak that I worked on. And again, I want to say how much I depended on this presentation and the information for this presentation from all of these folks. And just what a great job I think everybody did. And that is what I have. We're at 12:09, amazingly. And so if we have got any questions.
Great job, Elizabeth. Thank you. Let's see, we've got about 1 minute. So let's see, we'll go through, I'll pick a question here. One of the earlier questions that were put in the chat, were there any cases among staff that were involved in this investigation? You know.
So are we talking about riverboat staff or are we talking about staff as far as the folks who took samples? I would say certainly there were riverboat staff that were affected and did come up positive for norovirus. And none of our staff from Coconino County were positive.
Very good. Thank you for answering those because I wasn't sure and that responded included in the chat. So thank you very much.
You bet.
So for the sake of time, and there's a few more questions in chat, so I don't know, we may be able to get to those and answer those individually. But thank you. It was well received, Elizabeth. You've got a lot of thank you, great presentations, and a lot of comments there in chat. It was very interesting, and a lot of folks loved your humor as well. So thank you for sharing with us today.
Thank you. I appreciate you all so much.
Thank you. And up next, we're going to move right along to our next speaker. I would like to, our next speaker is going to be Dr. Liwan Yuan, and she is a full professor of virology and immunology at the Department of Biomedical Sciences and Pathobiology at Virginia Polytechnic Institute and State University, or Virginia Tech. in Blacksburg, Virginia. She received a PhD degree in virology and immunology at the Food Animal Health Research Program, Department of Veterinary Preventative Medicine from The Ohio State University. She completed her postdoctoral training at the Laboratory of Infectious Diseases at the National Institutes of Health in Bethesda, Maryland. She established her laboratory in 2007 at Virginia Maryland College of Veterinary Medicine at Virginia Tech as an assistant professor of virology. She has authored and co-authored over 100 scientific publications, including a recently book publication. She has received several awards, including the Pfizer Award for Research Excellence and most recently the Grant Turnwald Innovation Award from Virginia Maryland College of Veterinary Medicine. So without further ado, please help me welcome Dr. Yuan to the stage. All yours.
Thank you, Kenya, for the very nice introduction. So I'm a full professor of virology, immunology at Virginia Tech. My research heavily focused on norovirus and the rotavirus. So I'm very happy to be invited by FDA to give the seminar today talking about norovirus and my research. So here, to understand the norovirus biology, we need to talk about the naming, the structure, diversity, genotyping. I'm going to skip virus replication, the infection, the transmission, and pathology has been discussed earlier today, so I will just briefly mention it. I'm going to talk about the genetic resistance and the immunity and the notobiotic P model I use in my research. Then I will focus on the vaccine development effort from our lab and the landscape. I'm going to mute my video so we can all focus on the slides. Okay, so norovirus was first discovered in 1972 by Dr. Karpyshyn at NIAID. He collected a sample from a 1968 outbreak of gastroenteritis in a very small town. It's called Norwalk in Ohio. So Dr. Karpyshyn used the serum from the recovered patients and precipitated the stool sample looked under this electron microscope. So he found those small round virus-like particle. So he named the virus using the location where the virus was discovered. So it becomes the prototype of this genus of norovirus. So the naming convention for norovirus started with the species is human, and norovirus, most of the viruses are named based on the location where they are first discovered, and the year of the discovery and the country. Here, you see, with this exactly same electron microscope, Dr. Karpyshyn not only discovered norovirus, he also discovered another very important foodborne virus, the hepatitis A virus, together with Dr. Feinstein and Dr. Purcell. So let's look at the characteristics of norovirus. The virus are very small compared to other viruses. It's only 27 to 40 in diameter. On the surface, you can see 32 cup-shaped surface depression. So the genome of the virus is positive size single-stranded RNA of 7 kilobase pair long, contains 3 open reading frames. So inside of this protein capsid is this viral genome. This virus is a non-enveloped virus, not like SARS-CoV-2. We don't know the virus has a lipid envelope. This virus is naked, has no envelope. And the capsid is formed by a single major structure protein, is VP1. And here you see also VP2 is a minor structure protein. And the whole genome, you can see inside here, the purple part is it's a open reading frame 1. It encodes 7 non-structured proteins. Those non-structured proteins are important for the virus replication. And then the VP1 is encoded by open reading frame 2 is from this capsid protein. And then VP3 encode VP2 is a minor structured protein. Here, RDRP is important to mention because the sequence of the partial sequence of this genome is used to define the P type of norovirus. The sequence, complete sequence of this OV2, the VP1, is used to define the G type of the norovirus. I will talk about this later. Here you have a close look of the genomes from OV1 to OV3. And for the VP1, this capsid protein is divided into 3 different domains. The S domain is a shelf domain, and then the P domain, P2, inserted in the middle of P1 domain. And this P2 domain is hypervariable in sequence and is the primary domain that mediates the cellular receptor binding to initiate infection. And when we express this VP1 in a protein expression system, we can form different size of particles. If we express a whole VP1, we will get this virus-like particle is 180 nanomole, 37 nanometer in diameter. It's very similar to the native virus. And if we only express the S domain, we have this S particle is smaller, 20 nanometer in diameter. And if we express the P domain, the yellow part, the P domain only, we have a P particle in 2 sizes. If we have 24 more, we have this larger P particle. If we only have this 12 more, we have this small P particle. I'm talking about this here because it's important for the vaccine development. Vaccine has been developed using this VLP or this P particle as a vaccine antigen. So norovirus belong to the Calliciviridae family. So under Calliciviridae, there are 11 different viruses. You see the first one is norovirus. This norovirus is further divided into 10 different genera. Some of them infect human only, like G1. Some infect both human and pig. Some infect other animals. Here is the how we type norovirus. So as I mentioned, if we use the VP1 sequence, we call this VP1 typing, and we divide virus into genome groups. And under each genome group, there are genotypes. Then under each genotype, there are variants. And if we use RD-RP typing, we divide virus into P groups. And then also under P groups, we have P types. So from now, you can have the feeling about how diverse these norovirus are. There's so many different noroviruses. So this poses a very severe challenge for vaccine development. So here is a clear picture. So on either side, right side is VP1 typing and left side RDRP typing. We just look, see how this virus group is. So you see G2 group infect both human and infect pigs. G1 group only infect human. Some group like G3 only infect cattle, and G5 only infect mice. Among all these different genome groups, genotypes, the genome group 2 genotype 4 strain have been the most predominant strain over the past several decades. They account up to 85% of all the outbreaks. I was curious about the previous talk, what genotype was. In the recent years, in some geographic regions, some non-G24 strain has emerged and some even become predominant. The G24 synonym strain appeared in 2015. Actually, up to now, is still a predominant strain. G217 emerged in China for some years, and now we have this G22, G212 occurred since 2018. G24 norovirus is predominant across all age groups. You see here on the left side, so among all the age, there's this summary of from 55 different studies. G24 account for 62% of all the outbreaks. When you look at it in children less than 5 years of age, they also account for 67% of the outbreaks. Here is from the CDC CaliCNet genotype distribution from last September to this July. So you can see G2 Sydney P16 account for 43.4% of all the outbreaks. And there are many other genotypes account for less than 5%. This including G11. I mentioned here because G11 has been developed by several vaccine developers, but G11 actually now account for very small percentage of the outbreaks. So we heard about the transmission. Here, I just want to give some summary about why, the reason why this virus is so easy to transmit. The virus has a very, very low infection dose, it was mentioned, and also this virus has multiple routes of transmission. It comes from droplets for mice, environmental contamination, and there is a large human reservoir, prolonged asymptomatic shedding. Many people shed virus for long term without showing any symptoms, so pose risk for secondary spread, especially among the food handlers. This virus is a non-enveloped virus, so it's much more tough, more stable compared to enveloped virus. It's stable at relatively high level of chlorine and also temperature. From freezing to up to 60 degrees for 30 minutes, they can stay alive. So even in steamed oyster, you can still detect live virus. And also they are very resistant to extreme pH, from very low to very high for very long time. And as I mentioned, there is a very extensive strain diversity, genetic plasticity. So these multiple genetic and antigenic types lead to lack of cross protection. As we know, if we got infected by one type, we can be protected from reinfection for some time, but you can be affected by another different variant, different type of norovirus right away. So there's lack of cross protection, lack of long-term immunity and herd immunity. The virus infects gastroenteritis. It's not a systemic infection. So we know systemic infection can normally generate a very long-lasting immunity, but not the mucosal infection like norovirus and rotavirus, influenza. Those kind of infection only cause very short-term immunity. I want to here mention about the virus shedding titer, because this is very important to understand why the virus spread so easily, so widely. So people have a symptomatic infection after infection. We can detect the virus shedding range from 0.5 to 1 × 10⁹ viral particles from each grams of feces for 2 to 5 days. And this shedding actually can last for 2 to 5 weeks after the symptoms resolve. So from the previous talk, we saw there's a code about a restaurant worker. You are not allowed to come back to work after your symptom resolve for 24 hours. But actually, this is not enough. Those people can shed virus for very long. Especially if the people are immunocompromised, they can actually shed virus on and off for months and even up to years. So diagnostic kit is very important. We should all provide those sick people with at-home test kit, so they test themselves and make sure they stop virus shedding before they come back to work. Asymptomatic individuals shed high titers of virus actually similar to symptomatic individuals. Maybe also the transmission might be lower because they don't have this projectile vomiting and that spread virus everywhere, but they still shed virus from stool samples, and so it's a source of transmission. Rotavirus have a genetic resist factor. Some people are naturally resistant to norovirus infection. So there's early study in human volunteers, challenge studies. Dr. Kapikin led this study right after he discovered the virus. So the people was already inoculated with the human norovirus and stool filtration contained virus. Half of them got sick, half of them just nothing happened to them. 6 months later, and they challenged the same people again. The people got sick again. People did not get sick. Then now many years later, we realized actually the genetic factor is the histoblood group antigen. This antigen not only expressed on the red blood cells of blood typing, they are also expressed on the mucosal epithelial cells of human digestive tract. Norovirus infection use this HBG carbohydrate as a receptor is bound to the ligand to facilitate virus attachment to the virus. And this HBE antigen, H antigen, A antigen, B antigen, Lewis antigen, they are coded by this gene. It's called FAM2. It's found to code an enzyme, and this enzyme is important for expression of those HBG molecules on the mucosal surface. If an individual has this functional gene, they are termed secretor positive. They are susceptible to norovirus infection because they said the expressed HBG antigen on their mucosal surface can bind to the virus. People do not have this functional gene, they call secretor negative. They don't have those HBG antigens on their mucosal surface. They are naturally resistant to infection by most, but not all norovirus strains. Like in European population, 20% of people are actually secretor negative. Here on the left side, this diagram shows how this HBG mediated infection. So they expressed on the intestinal epithelial cells, and they brought the norovirus to the cell to initiate the infection. On the right side, this is just to explain what is blocking AC to measure antibody titers, and this antibody titer is actually used as a surrogate of neutralizing antibody. So on 96 valve place, we can coat this carbohydrate, and then the virus-like particle expressed in protein expression system and purified, and we can mix it with the serum sample to be detected all the monoclonal antibody we generate. And if there is a good titer antibody come into block, prevent this binding, and so we say there is a blocking titer. So there is we call it 50% blockage titer of this sample. So immunity to norovirus is not fully understood. There are some immune correlates for protection against norovirus infection in humans. Here is the list of this different type of antibodies has been associated with prevention, either with of clinical illness or infection. Here, one of them is the hemagglutination inhibition antibody. Here, diagram explain how this assay was performed. So the virus-like particle, if we mix with red blood cells, it can cause hemagglutination. So in the wells, you see this all red dot. But if there is some antibody can block this binding between the VLP and red blood cells, it prevents the hemagglutination. So we call inhibit hemagglutination. The red blood cells will precipitate to the bottom of the well. So you see a bottom, see a dot. This is there is a no titer. Here, there is a summary about the role of antibodies in norovirus immunity. So it has been reported that norovirus specific IgA, IgG, and fecal IgB titers did not correlate with protection. But pre-existing salivary IgA, serum IgBGA binding blocking, and IgA antibody titers are associated with a lowered risk and reduced severity of gastroenteritis. Some study reported if you have IgBA blocking titer greater than 200, IgA titer greater than 40, there is resistance to norovirus infection. A very recent study reported that heterotypic G2 serum IgB blocking titer did not prevent cross protection against illness from other G2 types. This is a very important study for vaccine development. So it's very clearly indicate that there is no cross genotype protect immunity. For vaccine to be effective, we need a multiple valent vaccine. Here is just the snapshot of this publication was earlier this year by Dr. Sylvia Decker. So antibody do not provide cross-genotype protection. And this is actually is complementing an earlier study done in 2014, have the similar conclusion. There is no cross-genotype protection, and the protect immunity is genotype specific and even strain specific. And the role of T cell response in norovirus immunity is in humans is totally not understood. We did some study in antibiotic pigs and we published the data. Here is just the only paper published earlier last year about norovirus specific CD8 T cell response, but they use healthy human donors and tissues. So it's not anything help understand the immunity against norovirus. So my lab using an animal model, antibiotic pig model to study norovirus. So what we can learn from using antibiotic pig model of norovirus, we can study what the infection dose, the pathogenesis, the HBG binding pattern, study immune responses, especially in some local tissue like in the gut, in response to norovirus infection or vaccination that's difficult to access in human volunteers. We can identify factors that affect susceptibility and replication of norovirus in this notobiotic, this clean background. Most importantly, we can use this model to conduct preclinical evaluation of prophylactics and therapeutics against norovirus. We all know during vaccine development, after the determining the antigen, the formulation of the vaccine, the most important first step actually to test the candidate vaccine in a preclinical animal model, and this animal model should mimic what human disease. So before we bring the vaccine to human clinical trials. So actually, the preclinical animal model serve as a gatekeeper. So you ask, what is notobiotic pigs? So the word gnoto is from Greek. It means known, and bio means life. So notobiotic pig is we know anything associated with the pig. Technically speaking, germ-free pig is also not about a pig because we know there's nothing, there are no microbes associated with this animal. The pigs are derived by surgical procedure 1 day before the expected due date, and they are kept in these isolators for the entire duration of the experiment. This isolator provides a sterile environment. It's totally free of any bacteria, mold, fungus, virus, etc., and except what we inoculate the pig with. Pigs also has no maternal antibodies because the cell close the uterus does not allow the transfer of macromolecules, unlike human and mice. So here, we have a very clean background. We can study true primary immune response to a single virus or a single vaccine in a very naive background. Here we contributed to the chapter 5 about notobiotic pigs from assembling facility to modern human infectious disease in this 2017 publication of notobiotics. And many people will ask, mice is much easier to much more available model to study. Why you use pig, not mice? First of all, mice cannot be infected by human norovirus. Mice can have their own norovirus, they call murine norovirus. This virus is widely spread among all the mice colonies. They replicate in the mice, and also we can culture them in vitro in macrophage and dendritic cells. But mice murine norovirus does not cause similar disease symptoms or tissue pathology to that of human infection in humans. Murine norovirus cause a systemic infection in immunocompromised mice, do not cause any disease symptoms in immunocompetent mice. So it's not a good model for study human norovirus. Notobiotic pigs are the only animal model suppose oral route of norovirus infection, develop diarrhea and shed virus in feces. And because the high similarity between and the pig and human in terms of their gastro physiology, the immune response, and the diet. The finding from pigs have high translational value to humans. Here I show two earliest publication using non-biotic pigs to study norovirus was conducted in Dr. Linda Safe's lab, who was my PhD mentor. So the first study showed that norovirus infect enterocyte of small intestine, unlike norovirus cause systemic infection, infect immune cells. Here in pigs, we only see the virus infect the small intestine. And you can see the continued infected cell, these blue dots are the norovirus antigen, some of them on the tip of the villi. Then the second paper found that the pigs actually very similar to humans. The secretor status A antigen positive, H antigen positive pig are much more susceptible to human norovirus infection than the A negative and H negative folk pigs. They're about fourfold different in terms of susceptibility. So from now on, all the study we use non-biotic pigs, we only use A positive and H positive pigs. And here is the first study I conducted in my lab, led by my student Tammy Bill. We determined the infection dose 50 of a G24 2006 strain. We found the virus shed very high in feces, and we see clear signs of diarrhea and cytopathological changes in the small intestine. And you can see here, this is the mark control pig, and the infected pig has this antigen detected in the damaged villi. And here at the electron microscopic imaging shows the signs of the cells apoptosis and the loss of the microvilli on the surface of the cells. And then we compared the ID50 we determined in the pig model. We found younger pig, newborn pigs has a lower ID50, and then the older pig will have increased ID50. And then we compared this ID50 to human challenge study. The norovirus in humans has ID50 of 2.8 times 10 to the third in all secret positive populations. And so our in pigs, we have a higher ID50. That means the pigs is less susceptible compared to this human virus in humans. With the model established, so we carried out several different studies. One is to see how the commensal bacteria influence infectivity. This bacteria, Enterobacter ecoli, was shown in vitro by other team to facilitate the virus infection of B cells. And so when we did the pig study, when we colonized the pigs with this E. coli, actually we found inhibition, not the promotion of norovirus infectivity. And we did not find any infected B cells. We did not observe infected B cells in any intestinal tissues in totally germ-free pigs or E. coli colonized pigs. And then we use immunocompromised pigs. Those pigs knocked out of two genes, and with this two genes knockout, the pigs do not have any B cells, T cells, or NK cells, so they totally immune deficient. And we found an increase and prolonged virus shedding in those pigs, indicating B cells, T cells, actually they're all important in immunity. And also we studied how human gut microbiota influence infectivity, because many studies have shown microbiota can enhance the infectivity of enteric virus. And indeed, we see colonized pig has enhanced the infectivity of human norovirus compared to the germ-free pigs. And the last study done by my student Shaohua is an efficacy study. He, you see here, treated pig. There's 5 groups with either a combination of 2 different probiotic strains, the Lactobacillus rhamnosus GD strain and the E. coli Nisi strain, in combination with raspberry and walnut. So the first group, we colonize the pigs, and then we start feeding the pigs raspberry, which is a prebiotic. 7 days before the challenge or 1 day after challenge. Control included the raspberry feeding alone, colonization with cocktail alone, or double negative. We observed very significant reduction in the percentage of diarrhea. Control pigs 89% with diarrhea, treated pigs only 20%. So the protection rate was 78%. This protection rate is higher than any vaccine has been tested in preclinical study and in clinical trials, actually. It's very promising approach for prevention of norovirus and diarrhea. We use the same model evaluate 2 different norovirus vaccines, and this is in collaboration with Takeda and also with Cincinnati Children's Hospital, Dr. Jason Jiang's lab. Takeda and also they changed former name was Lagoside. They developed this VLP and the P particle vaccine, so we tested in the pigs. I already talked about the P particle. The P particle is formed by the VP1 by the protruding domain, the P domain of VP1. The size of this P20 per particle is a very good size for subunit vaccine. So my student Jacob led this study, and he also studied how simvastatin, which is a cholesterol reducing drug, affects the protection and the T cell response induced by this vaccine in non-biotic pigs. Here we have 5 different groups. The first group of pigs were infected orally with norovirus and recovered. And the second group and third group are 2 different doses of P particles. And the next group is 100 microgram virus-like particles. They all use the same vaccine adjuvant as the Takeda vaccine. And the vaccine was given all intranasally. And we found after the virus infection, this is mimic natural infection, we see a high percent protection against diarrhea, 83%, and also 48% against virus shedding. In this 200 microgram P particle vaccine group, we saw 60% protection against both diarrhea and virus shedding. In the 100 microgram P particle or virus-like particle group, we saw 46% to 60% protection against the diarrhea, but not virus shedding. This image is the P particle antigen we use for vaccination. So the vaccine and the challenge virus are from 2 different G24 types. They are not identical in their epitopes, we call they belong to the same G24 genotype, but a different variant. So we call this cross variant protection. And then we compare the efficacy we saw in the pigs to human clinical trials. And this vaccine is a G11 VLP given intranasally, 100 microgram, has the exactly same protection rate as we saw in antibiotic pigs, 46%. And also protection rate against the infection is lower in humans. And bivalent vaccine in G11 plus G24 given intramuscularly also conferred a 46% protection rate against gastroenteritis, lower protection against virus shedding. And to further optimize our pig model, we did a series dose response study to determine ID50 and DD50. Another new challenge variant is norovirus G24 SIN2 variant. We give the pigs 7 different doses and then monitor the virus shedding, mean duration of virus shedding, and the diarrhea in the pigs. And we determine the optimal challenge dose is the group 3, is 2 times 10 to 5th. Here we see the group 3 pigs has the earliest onset of virus shedding, the highest, the longest duration, and the highest area under the curve of virus shedding, as well as the AUC of diarrhea. And we compare this information to what we see reported in humans. So humans given 5 times 10 to 4th of copy number of the virus, 70% of them shed virus, and the duration lasts for 5.2 days. Peak titer was 3 days. this is very similar to what we found in pigs when given 2 times 10 to 5th. We have 100% of shedding and 6.3 days shedding, and then the pig ate 4 days. We use various classical and novel mathematical models to calculate ID50 and DD50, and then we compare this ID50 with humans. We found actually it's pretty much the same, 2.8 times 10 to 3rd in humans, and we see ours in pigs 2.5. So this study is really very well standardized this animal model for preclinical trials. So this study attracted this article in Research Features earlier this year about this study. And in April, I published this book, Vaccine Evaluation, and this book includes how we establish the optimal challenge dose and different studies for using the pig model to evaluate human rotavirus and the norovirus. And then we started this collaboration with Indiana University. Dr. John Patton's group developed this platform using rotavirus as a backbone to express norovirus P particle vaccine. And this collaboration was funded by venture capital firm GIVax. We shared this $1 million grant. And here, I saw this food safety news. Probably this article is the reason I was invited to come here and speak today. So our goal is to develop a dual oral vaccine against both norovirus and rotavirus. So rotavirus is another important virus causing viral gastroenteritis in young children, infants and young children. Before there's a vaccine available, 500,000 kids die worldwide from rotavirus diarrhea. But now, since 2006 and 2008, there are 2 live attenuated vaccines become available. Now the vaccine available worldwide significantly reduce the disease burden of rotavirus, especially in developed countries. So this virus is also a non-enveloped virus. It has 11 segmented double-strand RNA, and the same is can replicate in small intestine, same as human norovirus, and the virus can induce strong systemic mucosal immune response. A reward genetic system for rotavirus was established in 2017. And we found the recombinant rotavirus can express foreign proteins, especially on the gene 7, which is ASP3 gene, does not affect the rotavirus viability and replication. A recent report from Stanford University, Dr. Greenberg's lab, reported that a rhesus monkey rotavirus expressed norovirus, the whole VP1, and when they infect 2 different types of suckling mice with this recombinant virus, they can induce VP1 specific antibody response. So here's a diagram shows the rotavirus reward genetic system. There are 11 gene segment, gene 7 encode ASP3. So we can insert this P domain into this gene and to express norovirus P particle. That's our goal. Then we'll evaluate this vaccine. So the ongoing key tasks, including we will use Rotarix strain to make this reverse genetic system, and we put this SIN2 strain P domain into this ASP3 gene. And then we recover the recombinant virus, and then we assess the genetic stability, amplification potential, suitability in culture in viral cell line. Then we study the immune response induced by this candidate vaccine in antibiotic pigs. Then we evaluate protective efficacy after we challenge the pigs with the SIN2 strain. And the next few minutes, I will just briefly go over the current landscape of other norovirus vaccine development. So here, there are several virus-like particle platform. The first one is the one we evaluate antibiotic pig. The company was first started with Leukocyte and then bought by Takeda, and now this vaccine is carried over by HelioVax for continued evaluation. The number in the squares are the number of clinical trials. So this vaccine has gone highest number of human clinical trials. Currently, it's finished phase 2. And another VLP, two VLP vaccine are both from China. They're expressed in yeast, and they are also carry already gone through human clinical trials. And we use pigs to study, evaluate this anhui vaccine. And the next three are the plant-based virus-like particle vaccines. And the last one, developed by Dr. Vaiskari in Finland, is a combination of G1, G24 plus rotavirus VP6. It has not started clinical trial yet. And there is a very attractive recommended an adenovirus vectored oral vaccine developed by VaxArt. Also has carried out clinical trials, and the mRNA vaccine developed by CureVac is studying in preclinical stage. Here, just a screenshot of the website from the company Helivax. They have showed how all the human clinical trials they have completed. The vaccine is a combination of G1 and G24, so it's bivalent vaccine, and it's given intramuscularly. So the vaccine is not oral vaccine, only include these two genotypes. And this is more detail about the anhui vaccine. It's a quadrivalent, has 4 different types. As I mentioned earlier, the virus is very, very diverse, and there's no cross genotype protection. So to have a broadly protected vaccine, we need multiple valent. So this vaccine is attractive because it included 4 different genotypes. Here, this is data from our non-antibiotic pig study evaluation. We saw the vaccine induced the antibody IgA, IgG titers across 4 different genotypes, and also induced good titers of hemoglobin inhibition titer before challenge and post-challenge. But after we challenged the pigs with the G26B strain, the G24 included vaccine is a Sundanese variant G24. The challenge virus is G24 2006B variant. We did not see good cross-variant protection of this vaccine because this intramuscular vaccine does not induce mucosal immunity. So this is a challenge for this vaccine. This is oral vaccine based on the adenovirus vector. It's oral vaccine, so I'm very hopeful for this vaccine. This vaccine has gone through all these clinical trials. But the challenge for this vaccine is also the most of the study was done with G11, but a recent clinical trial was a combination of G11 and G24, so it's just a bivalent. Okay, so here's a summary of the current knowledge about vaccine development. A number of vaccines has been developed, and those are based on VLPs and leading to the production of VLP or P particle in various expression systems. Preclinical early human studies of different concentrations and different adjuvants and various route of immunization, intramuscular or intranasal, and demonstrate the safety and immunogenicity. The intranasal monovalent G11 vaccine show protection 46% in humans, and also similar protective efficacy in our non-antibiotic pig study. But this intranasal route was abandoned by the vaccine company. They went to the intramuscular formulation. But when they challenged human volunteers, they only saw 13% protection rate based on this G24 challenge. And also, as I said, we did not see much cross-variant protection in our non-antibiotic pig study. There's some critical questions we need to answer. Can a vaccine be developed that elicit broad protection against multiple serotypes? So now enough study have tell us we really need a multiple valent vaccine. Okay, now one vaccine, single monovalent vaccine does not elicit broad protection. And will vaccine have to be updated regularly in order to match viral evaluation? Yes. Similar to the COVID vaccine, norovirus also involved, and the new variant emerge very regularly, so we need to update the vaccine. How will prior norovirus infection history affect vaccine immunogenicity and effectiveness? this is very hard to address in humans. Humans, 80% of humans got at least one norovirus infection before 5 years of age. Most likely multiple infections and with many different genotypes. So it's very hard to predict what in this kind of population, what kind of infect unless we carried out human clinical trials, phase 3 or post licensing monitoring. Will the same vaccine formulation to be effective in all age groups? My answer is no, because in human adults like greater than 5 years of age, the population already primed by the natural infection. When you give an intramuscular vaccine, this vaccine will serve as a booster dose, can increase antibody titer, actually reactive with memory B cells sit in the intestinal mucosal surface to provide protection. But for the infants and young children, if they have not been previously exposed by natural infection, intramuscular vaccine will not prime mucosal immunity. We need oral vaccine to prime immunity. And the last, how will the genetic susceptibility affect vaccine outcome? Is also needs clinical study to answer, because current study for the human clinical trial, they pretty much use only the secretor population. So we don't know how this affects the secretor negative population. And here's the summary of the key challenge we face. So immunity to norovirus is strain specific, genotype specific, no protection against genome groups, and very little protection across genotypes. So we need multiple valent vaccine. Genotype specific immune response and antigenic variation suggest multiple valent vaccine will be needed, and also require update when new strains emerge. Correlates of immunity is not fully understood. Some surrogates for protection have been proposed, but among all of this, the HBGA blocking binding titer is probably most close is associated with immunity. Intramuscular vaccine cannot prime for intestinal immune response in naive host. Pre-existing antibody to virus vector backbones also may affect immunogenicity of recombinant oral vaccine. Here, okay, so that's the last slide about vaccine. I will use the next 3 slides briefly talk about the development of passive immunotherapeutics against norovirus. The human monoclonal antibody against norovirus has been developed by Dr. James Cross lab, and also he formed this company. Another under development is IgY, chicken IgY monoclonal antibody developed by Scale Microbiomes here in Maryland, and also some company in China. So they immunize the hen with virus-like particle, and from the eggs, each of the eggs can contain roughly 100 micrograms polyclonal IgY antibody. This IgY can neutralize the G24P16 signaling strain. This actually developed was derived from another variation, the G24 variant. And so there is a cross variant protection against quite many years. And this IgY did not neutralize G26. So further confirm there is no cross genotype protection. And then the last is a nanobody developed from camelid from llama. It's from my collaborator, Dr. Viviana Perenio. Currently, she's from Argentina. Currently, she is a research scientist in my lab. So based on the nanobody she developed, we applied and received NIH R1 grant to develop probiotics yeast Saccharomyces boulardii secreted single domain antibody against norovirus. Those nanobody is also it's called just a heavy chain single domain antibody. They have some very attractive properties. Those antibodies very small, much smaller than the conventional antibody, and very easily soluble. And they have some crypt epitope can bind to the inside of the various particles. They are very stable in harsh conditions, such as extreme pH and temperature. And they have good tissue permeability, and also easy to produce. They can be made in E. coli. So we use this probiotic yeast because it has a long history as effective diarrhea-reducing probiotics. Here, that show that we have 3 different nanobodies. We insert them into human IgY Fc segment, and we form this type of bivalent homodimer or bivalent heterodimer of the structure. Or we can even make the 4 different type of the structures. And then the ongoing study of this project, we first confirming if this structure can colonize antibiotic pigs and secrete high level of antibody. We actually, as of today, we just euthanize the pigs, and we saw the antibody production in the intestinal contents of antibiotic pigs. And then we will determine the protective efficacy against norovirus diarrhea when we challenge the pigs fed of this probiotic yeast. So here is an acknowledgement. My research mostly funded by NIH and also by the vaccine company, and now currently also by GI Vax. This is currently my lab member. That's my last slide. Thank you for your attention. I will stop sharing here now. Okay, I'm done.
Thank you so much, Dr. Yuan, for your presentation. I'm super glad you were able to work with us on our technical difficulties once again. Unfortunately, we don't have time for any questions, but we're going to stick around and we'll try to answer some that are in the chat if we can. But I don't know if you see the chat box, but you're getting a lot of thank yous. So thank you very much once again for presenting for us today. We're so excited that we could have you here.
Thank you.